Hello students, welcome to EPG Patashala. I am Gaurinath from School of Life Sciences, Jawaharlal Nehru University. Today we are going to discuss about a module on protein-protein interactions and structural perspective. So mostly we are talking about structural perspective of protein-protein interactions. Before going to the structural part, I will introduce you to several biochemical methods used in protein-protein interaction studies, and then we will see the examples of protein-protein interactions, protein-protein complex structures, and from these structures, what do we understand the common features in protein-protein interactions. So, as I said before, there are several ways one could study the protein-protein interactions. The simplest one is spectroscopy methods, with simply the absorption spectroscopy or fluorescence spectroscopy, where if the proteins interact with each other, the interacting residues are involved with hydrophobic residues like tryptophan and tyrosine, the absorption will change or the fluorescence of these residues will change. We can also introduce chromophores, and if the chromophore is at the site of protein-protein interactions, the fluorescence signal due to this chromophore will change. So when they are exposed to the solvent, their absorption or fluorescence signal is different, and when the other protein binds, the environment of this chromophore or the residues like tryptophan or tyrosine will change, their spectroscopic properties will change. So with this one could understand the protein-protein interactions. The other way is dynamic light scattering. Here basically if two proteins interact with each other, the size of the resultant molecule will change. So the dynamic light scattering, we can measure the radius of a particle. So if you have two independent particles, you basically should get two peaks, and when these interact, the resultant peak should have a higher radius and single peak. Similarly, we can do a column chromatography. So if two proteins interact with each other, they form a stable complex. So elution volume of the complex should be different than the two individual proteins. You could do a surface plasmon resonance where you immobilize one protein, pass the other protein with the surface plasmon resonance signal. We could understand whether the protein is binding or not, and this is more sensitive where you could measure the binding affinity of these two proteins. The other ways of studying the protein interaction is ultracentrifugation. It's very similar logic as a column chromatography or dynamic light scattering. If the proteins interact each other, the larger complexes form, their sedimentation velocity will be different than the individual components. You could do yeast to hybrid system where in the yeast the two proteins are ligated with different proteins, and when they interact, then you should have a different signal, and that will give you information whether these two proteins interacting or not. So there are several ways of studying protein-protein interactions, but all these methods generally tell you whether these two proteins interacting with each other or not, except some cases the fluorescence and absorption spectroscopy, maybe you can even tell which residues are involved if the experiments are successful. Otherwise they generally tell you whether a protein A and B can interact with each other or not, but they won't tell you what residues of protein A and what residues of protein B are interacting. For this, the complex structure determination is only the best way to understand these protein-protein interactions. If you look at the structures in the PDB, there is a robust growth of structures determined in last several years. The growth is almost logarithmical. If you look at today, we have about 1,20,000 structures in PDB. Now if you look at these structures and pick up the protein-protein complexes, the structural complexes, one could see what kind of interactions and how these proteins are interacting with each other. Now can we learn from the structures what is the mode of interactions, and from there can we extrapolate to predict, if we know the structures, whether what kind of proteins interacts with which kind of proteins. So on the basis of these structures, one could classify into broadly 4 classes. Now why only these 4 classes? Because there are not many structures which could be classified into many different ways. So earlier the structures were determined mostly from natural sources. So all the structures which we have, the protein complexes, are either antigen antibody complex or enzyme inhibitor complexes. There are many proteins which forms oligomers like dimers, trimers, tetramers, hexamers, etc. So the interaction between these two protein monomers is also the example of protein-protein interactions. The last one is the cellular proteins. The cellular signaling proteins need to interact to give a cellular signal in the cell physiology. But till recently, there are not many structures in this class. But now, as the improvements in the molecular biology techniques in last decade, there are several structures which have been reported in this area. Now, of course, there is not enough work has been done to classify the protein-protein complexes in this area. Now in this module, I will try to tell you examples of protein-protein interaction structures from each of these classes. Here I am showing you the example of antigen-antibody interactions. We can see the large antibody and a small antigen in this structure. Now, generally, antigen-antibody interactions, the binding region is very small. So one could see the large complex structures where you have a large antigen and antibody, but the binding region is very tiny. So if you see the zoomed picture of antigen binding site to the antibody, you can see the green one, which is from the heavy chain, the light colored protein, which is from the light chain, and there is a blue colored structure which is shown as a ball and stick model, which is antigen. The yellow color residues are from the antibody. Now these interactions are mostly the combination of hydrophobic interactions, and we can also see some hydrogen bonds and hydrophilic interactions, salt bridges. All these together form this antigen antibody interactions. The variable loops between the light chain and heavy chain loops, which causes the variation of antibody and changes in the specificity. The residues from the peptide from the specific stabilizing non-covalent interactions with the residues from the hypervariable loops of the antibody. These unique set of interactions allow the antibody to bind antigen with very high specificity. The multimeric proteins, we can see many structures in the PDB. Here I have shown you some of the multimeric structures which are determined from my lab. The phosphoglycerate dehydrogenase from Entamoeba histolytica, which forms a strong dimer. You can see the two molecules shown in two different structures, and there are two helices in the zoomed picture. You can see these two helices, which are shown as the cylinders, are very close to each other, and there are several hydrophobic residues which are forming hydrophobic pack interactions. At the peripheries, you can also see several ionic interactions and hydrogen bonds. So these interactions which makes this stable dimer. The PGDH, if you see in charged base color format, you can see the dimeric interface which is completely white. So the white is basically the hydrophobic interactions, and the blue colored is the positive charge residues, and the red ones are the negative charge residues. So in the dimeric interface, you can clearly see it's dominated by a white color, clearly indicating there are several hydrophobic interactions. So in this structure, you have 10% of the total surface area which is involved in the dimeric formation. The structure shows the electrostatic hydrophobic interactions at the dimer interface play a crucial role in the stability of the protein, and the complete dimer is essential for requirement of optimal enzymatic activity. If we disturb the dimer, these enzymes become non-functional. As I said before, the hydrophobic interaction in the center are a dominating one which stabilizes the dimers very strongly. Here I am showing another example of multimeric protein structures. Here I am showing you another structure determined from my lab is a calcium binding protein 1 which forms a trimers. So it's an elongated structure. The EF1 motif and EF2 motif are connected by long helices. But in this trimer, the EF2 of one molecule interacts with the EF1 of another molecule and forms an assembled domain. This assembled domain is very similar to Calmodulin single domain, and this is similar to a domain swapping manner. Here it's a motif swapping which happen and which causes the calcium binding protein 1 in trimeric form. If you zoom into any of the one domain, and you can clearly see in the center the interactions are dominated by hydrophobic interactions. You can see the phenylalanine from two molecules, the isoleucine and tyrosine from two different molecules, the valines isoleucine from two different molecules come together to form hydrophobic patch. Then you can also see several hydrogen bonds between the two molecules and the ionic interactions at one corner where the lysine and glutamic acid from two molecules to come and form a salt bridge. So it's a combination of several ionic and non-ionic interactions and mostly dominated by hydrophobic interactions, which makes these motifs come together to form assembled domain. So the second most common example of protein-protein interaction is enzyme inhibitor complexes. In olden days, most of the proteins are extracted from the natural sources. So in the literature or in the PDB, if you look at, you get a lot of these enzyme inhibitor complexes. And the most common one is serine proteases. So here I'm going to describe a little bit about the serine proteases and how it is complexes are there in the literature and how they interact. The serine proteases can hydrolyze either ester or peptide bond utilizing mechanism of a covalent catalysis, where the intermediate state intermediate state is after cleaving the peptide bond, one peptide is still bound to the serine of the enzyme and other fragment is released. So serine proteases play an important role in many processes like digestion, blood clotting cascade, and several pathways, differentiation and development. So classical examples of serine proteases are trypsin, chymotrypsin, and elastase. Here I am showing you the structure of trypsin. The trypsin is dominated by beta sheets. So there are only two small helices at the periphery and most of the structure is with the beta sheets and several loops. So you can see there is a small cleft in the center and there are several beta sheets and loops. In the next slide, I will show you the space fill model. Bovine pancreatic trypsin inhibitor is one of the best inhibitors where the trypsin and BPTI complex structure has been solved quite some time back. This binds to trypsin as a substrate and forms an acyl enzyme intermediate rapidly. So when it binds and after acyl enzyme intermediate complex formation, there is very little structural changes in the trypsin or BPTI. we can see the BPTA structure, which is very small protein, which has just two antiparallel beta sheets and two small helices and mostly loops. It has several cysteines which keep the structure very compact and tightly packed structure. So in the complex between the trypsin and BPTI, there are several hydrogen bonds between the backbone of two proteins. So the complex is so strong that the hydrogen bond between the backbone of two proteins all forms very similar to antiparallel beta sheet, and this causes the binding very strong. Now BPTI, which mimics very similar to substrate, rather it's a very good substrate. Now once the trypsin and BPTI binds, then why it doesn't hydrolyze? Rather, the hydrolysis, the cleavage of BPTI is very slow. Now this is because the water molecules do not have access to the active site. Now because there is no water molecules close to the active site, there is no hydrolysis. The tight binding of trypsin and BPTI is responsible for not having access to the water molecules. Even after the cleavage of the peptide bond with the active site, that is new acyl complex formation, as there is no water, there is no hydrolysis. And this acyl enzyme intermediate is in close proximity with the cleaved peptide. Now this causes a reversible reaction where the peptide bond can reform. The rigidity of BPTI may also contribute by not allowing necessary atomic motions for hydrolysis to happen. So the tight binding and the very rigid substrate structure are another responsible components for non-hydrolysis. Here I am showing the structure of trypsin BPTI complex. The BPTI active loop, which is substrate mimicking loop, which enters into the active site of trypsin. You can also see in the zoomed picture where the lysine from the BPTI interacts with the aspartic acid of the specific pocket in the trypsin, which is responsible for specificity of trypsin. Once this is interacted, the active site can cleave. But in the case of trypsin BPTI, or even after all favorable interactions and binding, the trypsin cannot cleave. If you see the next slide, you will understand why there is no access to water molecule. The same trypsin BPTI complex structure is shown in the space fill model. The red color structure is trypsin and cyan color, a bluish color one, is BPTI. If you see the structure, there is no space between these two structures. The binding is very tight. The complementary of the surface structures is very accurate. So the surface complementarity and the rigid structure of BPTI, which makes the complexes very strong. As we have already discussed, there are several hydrogen bonds are formed between these two structures. The charges are opposite to each other, where the lysine from the BPTI and aspartic acid from the trypsin, a negative positive charge of the active site of trypsin and the loop of BPTI. Everything is complementary to each other, which makes this complex very strong and stable. So far, we have discussed about different protein-protein interactions. We have discussed antigen-antibody, the multimeric proteins, protease inhibitor complexes. Now I will show you few cellular proteins which form a protein-protein complexes. The best classical example is cyclin-dependent kinase and cyclin complex. The cyclin-dependent kinase, which is we call it as a CDK, and cyclin complex structures are well studied. Now kinase function as the switches to regulate the transition of the cell from one phase to another phase. So cyclin dependent kinase, which is actually a switch which regulates the cell from changing from one phase to another phase. CDKs are the smallest known members of protein kinase family. So they are very small proteins which are regulated. The function of this kinase is regulated by cyclins. Now in the next slide I will show you how it influences the cell cycle. Here you can see the cell cycle. Most of you know the cell cycle very well. The G1 phase, S phase, G2 phase and M phase, which gives the 4 phases of the cell cycle. Now this is one regulated by cyclin and cyclin dependent kinases. In the beginning of G1 cycle, the cyclin dependent kinase is expressed, which is non-active. In the middle of G1 cycle, the specific cyclin which is expressed and which binds to this cyclin dependent kinase. Once it binds, it activates this kinase. Once the kinase is activated, it starts phosphorylating several proteins, indicating that it should now change the phase to S phase. So that's how it pushes the cell from G1 to S phase. Once it enters into the S phase, this complex is degraded. Now in the S phase, as we know, the DNA is duplicated and several things takes place. The cell size increases. Now at the end of the S phase, you have another set of cyclin dependent kinases are expressed. Now in the beginning of G2 phase, again they are not active. In the middle of the G2 phase, when specific cyclins are expressed and they bind to again the cyclin dependent kinases and start phosphorylating several proteins, and which pushes the cell from G2 phase to M phase. And as soon as it enters into the M phase, then this complex is degraded. Now that's how the cyclin dependent kinase and the cyclins which regulate the cell cycle. Now the complex between the cyclin dependent kinase and cyclin is very interesting, and here we can not only study the protein-protein interactions and how the structural changes are induced in the cyclin-dependent kinase, which is changed from the inactive conformation to active conformation, can be studied with this example. In this slide, I am showing you both independent structures. The cyclin-dependent kinase, we can see the structure. It has a large C-terminal domain which is full of alpha helices and a small N-terminal domain which is full of beta sheets. These two domains are connected by two loops. One is called T loop, another one is called PSTAIRE helix. This PSTAIRE is basically the sequence of the helix which is made up of these residues. So that is proline, serine, threonine, alanine, isoleucine, arginine and glutamic acid, which is made up of this helix. These two are very flexible in nature. If you look at the cyclin is alpha helix rich protein which you can easily can be divided into two small domains. Those two domains are very identical with each other, which is also expected. This is in evolution, it is due to the gene duplication event. The cyclin is mostly rigid structure. The cyclin dependent kinase is shown in two structures state. One, the CDK, which is inactive state when it is not bound to cyclin, and the other which is bound to cyclin in an active conformation. The active conformation, only the cyclin dependent kinase is shown and cyclin is not shown, which clearly shows the structural changes in CDK from inactive state to the active state. In the active state, you can clearly see there is a white patch on cyclin dependent kinase which is kind of exposed. And that is the one which is actually interacts with the cyclin. It is not exposed to the water, it is actually exposed to the cyclin. In this structure, I have shown you the complex structure of CDK and cyclin. We can see that when the cyclin is bound to CDK, there is the structural changes which I have showed you before to the N-terminal domain, and this complex is very stable. In the right side, the hydrophilic residues which are shown in the center are actually nothing is coming at the interface. At the interface, it's mostly hydrophobic interactions. In the next slide, I'll show you the charged surface structures. Here, the cyclin and cyclin dependent kinase are shown in charged dependent color format. So the boxed area, which is actually the protein interacting surface, we can see that the white patch is larger on both cyclin and cyclin dependent kinase. So these white patches which interact with each other and form the complex. You can see the complex to the right side. So basically, the hydrophobic patches on the both molecules which interact with each other and form this complex. In this slide, I am showing the conformational changes in cyclin dependent kinase. The left part, where cyclin is not bound, you can see the orientation of PSTAIRE helix and T loop. When it is bound to cyclin, the PSTAIRE helix is pushed and it is rotated at the glutamic acid level. And the T loop, which had a small helix, which is actually changed into extended structure and which results in a small beta sheet when bound to cyclin. So there is a huge structural change in these two loops when cyclin is bound to kinase. We can also see the two structures together when they are superimposed on each other at the right side of the slide. Those two structures again zoomed at the interacting surface on kinase. You can see the PSTRI helix, which is not only moved and it's also rotated, and you can also see the T loop, which converted the yellow colored part, which is a T loop, which becomes extended structure. It's long loop. There are three arginine residues near the T loop which are dispersed in different orientations. You can clearly see there is arginine on PSTRI helix and near the catalytic loop there are two arginines which are differently oriented. Naturally, all of them are positive charge residues. Now the T loop, which has a threonine, this actually plays another important role in active conformation. When the cyclin is bound, there is a huge structural change which is stabilized by the phosphorylation of threonine, and that causes the interaction between these arginines and the phosphorylated threonine. In the next slide, I will show you how these interactions are formed. So when the cyclin is bound to kinase, it also causes autophosphorylation. So the threonine, which is phosphorylated, becomes negative charge, and the arginines from the surrounding region interacts with the negatively charged phosphorylated threonine, and this causes the stabilization of structural changes in the kinase. So the kinase not only helped by moving this due to movement of several parts as the cyclin is bound, is also the conformation is stabilized by autophosphorylation and the interaction of phosphorylated threonine with several arginine residues. Here I am showing you the kinase in the space fill model. The color format is still maintained. The yellow color one is the T loop, and PSTRI helix is shown in the brown color. So in the inactive state, the T loop is actually covering up the active site. But when the cyclin is bound, the T-loop conformation is changed, it's moved away. The ATP binding site and the active site of kinase is now open. So in the inactive state, we can clearly understand it cannot bind to substrates because T-loop itself covering up the active site. And not only this, the glutamic acid, which is essential for hydrolysis of ATP in the ATP hydrolysis and phosphorylation. So the structural changes which moves away a T loop, which opens up the active site complex and also has caused changes in the PSTRI helix. The glutamic acid now which orients in a proper orientation, which can do now ATP hydrolysis. The another example of uh, protein-protein complexes is cellular proteins, and Calmodulin is one is a classical example. Highly regulated changes in the concentration of cytosolic calcium ions control several biological processes, very diverse biological process like muscle contraction, fertilization, cell proliferation, vesicular fusion, apoptosis. And many of these cellular effects are modulated by small changes in the calcium concentration through calcium binding protein calmodulin. The calmodulin is 15 kilodalton small protein, a soluble protein, and if you look at the evolution, calmodulin is highly conserved among the vertebrates. It has just 15 kilodalton domain protein, which is divided into two domains. Each domain has two affine motifs connected by long helix like a dumbbell shape. I will show you the structure after a couple of slides. The calmodulin binding targets are diverse in their sequence. Even though the calmodulin binds to helical kind of structures, but the sequence of the helix need not to be same. So different kinds of targets it binds to. So I am going to explain all this examples in this module. The CAM binding proteins are diverse group of targets whose interaction with CAM can be subdivided into calcium dependent and calcium independent modes. Some of the target proteins are the ones which I am going to explain here are calcium dependent ones are CAM kinase, myosin light chain kinase, myosin liver arm which interacts with myosins, nitrous oxide synthase, IP3 receptor and calcium pump. So these several kinds of these proteins, the functions are completely different, their binding site sequences are different, but calmodulin binds to these proteins and change their function. The Calmodulin structure you can see here, it's almost like a dumbbell shaped. The two affine motifs which forms the N-terminal domain and another two affine motifs which form the C-terminal domain, which are very close to a globular structures which are connected by a long extended helical structure. This dumbbell shaped structure which has a opens up the hydrophobic cleft in each domain now ready to bind to targets. When it binds to target, the central helix is melted and it wraps up the target helix. Generally, the calmodulin binds to a helical region of the target which stabilizes the helical conformation, and it binds to two hydrophobic residues at the two ends. Here, the example is shown as CAM kinase 2 CAM complex structure where two hydrophobic residues are entering into two hydrophobic clefts in the calmodulin. The calmodulin binding to its target could be very flexible. Now the central helix is melting and it has to wrap up the target helix. Now these could be quite different, like it can bind to 1,10 motif a 114 motif, a 116 motif, or IQ motif. So what do you mean by 110 motif? So the first residue and the 10th residue has to be hydrophobic residue. So where the first residue and 10th residue, the Calmodulin hydrophobic pocket goes and binds. Now the central helix is melted, now it can reach different lengths. In the case of 114 motif, it binds to first and 14 residues. It can also bind to 116 motif, where the first and 16th residue is a hydrophobic motif. Now these motifs are further stabilized by several changes. Like example, they could be preceded by positive charge residues. We know that calmodulin is a negative charged molecule. Other than these hydrophobic interactions, if we have positive charge residues preceding the motifs, it can have a favorable interactions. Now these motifs could have another hydrophobic residue somewhere in the center. When the helix is melted, it could make a favorable hydrophobic interaction between the melted helix of calmodulin with the target. The IQ motifs is generally seen in the unconventional myosin, where IQ motif I is isoleucine followed by glutamine. Now, it has also a similar pattern of residues, where the first residue is isoleucine and it has a specific pattern of residues where the example is sixth is a glycine, etc., and has another hydrophobic residue at the end. The calmodulin can also bind to other targets in non-canonical model, where it can have 1:2 or 2:2. So 1:2 is where one target is bound to two calmodulins, or 2:2 is the two targets are bound to two calmodulins. The 2:2 classical example is calcium gated potassium channel, where two calcium gated potassium channels are bound with two calmodulins and reorients the channel in active conformation. So if you look at all these structures which we have talked about, protein-protein complexes, we can conclude or we can extract a few prominent points. What makes these protein-protein interactions more favorable? So the first, if you look at the surface accessibility area, what kind of surfaces these proteins form? Do they have a shape complementarity? Or if they don't have a shape complementarity, the binding, the interaction should be favorable enough to change structural conformation and cause the shape complementarity and make a favorable interactions. And third is interface residues. Even if you may have a shape complementarity, but the residues, the atoms which are interacting should have a favorable interactions. Generally, we have seen the interactions are dominated by hydrophobic interactions, which are further stabilized by hydrogen bonds and salt bridges. Now, in the physiological condition, most of these proteins are in water. So the hydrophobic patches on the protein surfaces cannot survive by itself. They look for their partners where these hydrophobic patches could be covered. So that's one of the primary indication the protein, if they have hydrophobic patch on the surface, it's ready to interact with the another protein. Now we summarize this module on protein-protein interactions. As we discussed before, the protein-protein interactions could be studied by different biochemical methods. All these methods will only suggest the interaction between two or more proteins, but they do not exactly specify or suggest the interacting regions or interacting residues. Today in PDB we have 1.2 lakh structures, and more protein complex structures are getting reported. From these complex structures, we could broadly classify them into 4 different classes. Here we have discussed about the examples from all the 4 different classes, and from these protein-protein complexes, what could we understand? The protein-protein interactions basically occurs, the minimum necessities is surface complementarity. So the protein of one shape, or you need to have a complementarity shape to interact. You cannot have different shape ones. So the surface complementarity is very important. The interface residues play a major role. Like generally you need hydrophobic residues which play a role in protein-protein interaction. Many of the cytosolic proteins, they don't like hydrophobic residues on their surface. So any protein which has hydrophobic residues on the surface, they look for a counterpart protein which has a surface complementarity, and they interact. So on the basis of this knowledge, if the structures are known, one could predict the interaction between two proteins and their interaction sites. So one, we have learned how this protein-protein interaction takes place. Now we could also guess and predict if one independent structure is determined in the protein structure database of 101,20,000 structures, can this protein interact with any of other proteins? So there are websites, methods available to predict these protein-protein interactions.